Good morning, everyone, and welcome. I'm delighted to have you here today. My name is Dr. S.K. Singh, and I hold a PhD in this field. Today, we'll be exploring modern approaches in sterility testing. In this presentation, we'll look at the latest techniques and innovations that are shaping how we ensure sterility in various products. We'll cover key methods, recent advancements, and best practices. I'm excited to share this knowledge with you, so let's get started. Welcome to our discussion on sterility testing. So, what exactly is sterility testing? It's a process that confirms the absence of viable microorganisms in a product, ensuring that the product is free from any living contaminants. This step is not optional. It's mandatory under several key standards and regulations. For example, the United States Pharmacopoeia, or USP 71, and the European Pharmacopoeia, Section 261, both specify requirements for sterility testing. Additionally, the FDA's Regulation 21 CFR 211.167 emphasizes its importance in pharmaceutical manufacturing. Now, why is this so critical? Well, sterility testing plays a vital role in product release. It's one of the final checks before a product reaches the patient, ensuring safety and compliance. Without it, we risk releasing products that could cause infections or other adverse effects. Importantly, sterility testing is a core element of good manufacturing practice, or GMP, microbiological requirements. It supports our commitment to quality, safety, and regulatory adherence. So, in summary, sterility testing isn't just a routine step. It's a fundamental safeguard in delivering safe, effective products to patients around the world. Now, let's look at how modern approaches to sterility testing are classified. We have three key methods that are widely recognized today. The first is membrane filtration, which is actually our preferred compendial method because of its reliability and efficiency. Then there's direct inoculation, which serves as an alternative compendial method when membrane filtration isn't suitable. And finally, we have rapid sterility testing systems, often referred to as RMM, which are becoming increasingly popular due to their speed. When we compare these approaches, they differ based on several factors. For example, the detection mechanism, how each method identifies microbial presence, can vary significantly. Sensitivity is another critical point. Some methods can detect lower levels of contamination more quickly. Time to result is also essential, especially in fast-paced manufacturing environments where quicker results can lead to faster decision-making. Lastly, regulatory acceptance plays a big role in determining which method is appropriate for a given application. Understanding these differences helps us choose the most suitable approach depending on the specific needs of our sterility testing process and compliance requirements. Now, let's take a closer look at the principle behind membrane filtration. Essentially, it's a physical separation method that uses a membrane with a pore size of 0.45 micrometers or smaller. This allows us to separate microorganisms from the sample effectively. The process begins with filtering the sample through this membrane, which traps the microbes on its surface. After filtration, we rinse the membrane to remove any substances that might inhibit microbial growth or detection. Once the microbes are retained and the inhibitors are washed away, the membrane is incubated in specific media like FTM and SCDM. This incubation step is crucial because it supports the growth of any trapped microorganisms, making them easier to detect. An important point to highlight is that this method not only isolates the microorganisms, but also helps remove inhibitory substances. By doing so, it significantly improves our chances of accurately detecting and identifying the microbes present in the sample. Overall, membrane filtration is a straightforward but powerful technique that enhances the sensitivity and reliability of microbial detection in our testing process. Now, let's take a closer look at membrane filtration from a regulatory perspective. The preferred methods for this process are outlined in USP 71 and PH, UR 261, which are the key guidelines we follow. These methods are required when the product is filterable, meaning if your product can pass through a filter, you should be using membrane filtration. From a regulatory standpoint, it's essential to demonstrate the integrity of the filter, showing that it's working properly and effectively. Additionally, you need to validate rinsing efficiency, ensuring that the filtration process doesn't leave behind contaminants or residues. Performing method suitability is also critical, meaning you need to confirm that your filtration method is appropriate for your specific product. Regulatory bodies tend to favor filtration because it offers higher sensitivity compared to other sterilization or sterilizing filtration methods. This higher sensitivity helps ensure the safety and purity of the final product, which is why it's often the preferred approach in the industry. Let's take a closer look at membrane filtration and its advantages and limitations. First, on the advantages side, one of the key benefits is its high sensitivity. This method can detect very low levels of contaminants, which is crucial in many testing scenarios. Additionally, membrane filtration is effective at removing inhibitors such as antibiotics and preservatives, which can interfere with other testing methods. This makes it particularly useful when working with complex samples. It's also well-suited for testing large volumes, allowing us to process more sample material efficiently. Now, turning to the limitations, there are some types of products where membrane filtration isn't ideal. For example, oily products and highly viscous formulations can pose challenges because they don't pass through the membranes easily or uniformly. Another important point is that membrane filtration requires specialized equipment, so it's not always practical in every setting. Recognizing these limitations helps us decide when this technique is most appropriate within our overall testing strategies. Now, let's talk about the principle behind direct inoculation. Essentially, this involves adding a sample directly into specific culture media to check for microbial growth. The key idea here is the direct addition of the product into the media, which allows us to observe whether microorganisms are present. The procedure involves inoculating the sample into two types of media. First, we have the fluid thioglycolate medium, which is incubated at around 30 to 35 degrees Celsius. This medium is excellent for supporting the growth of anaerobic and aerobic bacteria. Second, there's the soybean casein digest medium, which is incubated at a slightly lower temperature, between 20 and 25 degrees Celsius, to support different types of microorganisms. The incubation period typically lasts about 14 days. During this time, we observe the media for signs of growth, mainly looking for turbidity, which indicates bacterial proliferation. Increased turbidity means the microbes are growing, and this is a crucial indicator in our analysis. So, in summary, direct inoculation is a straightforward yet powerful method to assess contamination, and by observing turbidity after incubation, we can determine if microorganisms are present in the product. This approach ties into our overall theme of reliable microbial detection in quality control. Let's take a closer look at the regulatory expectations around direct inoculation. First, it's important to understand that a justification is always required. You need to clearly explain why you're choosing this method. Additionally, the method's suitability is mandatory. You must demonstrate that it fits the specific situation. When it comes to direct inoculation, the regulatory position is quite clear. It's allowed only when membrane filtration isn't feasible. For example, this might be the case with oily products, where filtration can be challenging, or with medical devices, which often have complex materials. Insoluble materials also fall into this category because filtration might not be effective or practical. It's also worth noting that direct inoculation is generally considered secondary to filtration. That means, whenever possible, filtration should be the primary choice, and direct inoculation should be used only when filtration isn't a viable option. This approach ensures we meet regulatory standards while maintaining the integrity and safety of our processes. Let's take a closer look at the limitations of direct inoculation. One key issue is that it doesn't involve removing inhibitory substances that might be present in the sample. This can affect the test's accuracy because those substances could interfere with detection. Another important point is the risk of false negatives. Since we're relying on direct inoculation, we might miss some true positives, especially if the sample contains inhibitors or low levels of the target. This approach generally has lower sensitivity compared to filtration methods, which can concentrate the target and improve detection. It's also crucial to highlight the strong dependence on neutralization validation. Because the success of direct inoculation hinges on neutralizing potential inhibitors, proper validation is essential to ensure reliable results. Lastly, product interference remains a major concern. Components within the sample or the product itself can interfere with the test's performance, leading to inaccurate outcomes. So, while direct inoculation can be useful in certain scenarios, it's important to be aware of these limitations to interpret the results correctly and consider alternative methods when necessary. Now, let's take a closer look at rapid sterility testing systems, or RMM as they're often called. These are essentially alternative validated methods designed to provide faster microbial detection, which is crucial in maintaining product quality and ensuring timely release. Instead of waiting days for traditional culture methods, RMM offers a more efficient approach. Some of the key technologies used in these systems include ATP bioluminescence, which detects microbial activity by measuring light emitted from ATP, the energy molecule in all living cells. Then, we have PCR-based detection, which amplifies microbial DNA to identify contaminants quickly and accurately. Flow cytometry is another advanced technology, allowing us to analyze individual cells rapidly, giving immediate insights into microbial presence. Lastly, CO2 detection systems monitor the production of carbon dioxide as an indicator of microbial growth. As examples, systems like Milliflex Rapid and BACT/Alert are widely used in the industry. These tools help streamline testing processes, improve sensitivity, and reduce the time needed to ensure sterility. Overall, integrating RMM into our quality assurance programs aligns with our goal of faster, more reliable microbial detection, ultimately supporting better product safety and compliance. Now, let's look at the regulatory framework that supports rapid systems. First, we have USB 1223, which focuses on the validation of alternative methods. This provides a solid foundation for demonstrating that these new approaches are reliable and fit for purpose. Then, there's the FDA PAT guidance, which specifically addresses process analytical technology and encourages innovative, real-time monitoring solutions. And of course, the EU GMP Annex 1, which sets the standards for sterile manufacturing and actively promotes the adoption of rapid microbiological methods. So, what are the key requirements here? Well, first, you need to demonstrate equivalence or even superiority of your rapid methods compared to traditional ones. This is crucial for regulatory acceptance. Second, performing validation studies is essential. These studies provide the evidence needed to show your methods are accurate and reliable. It's interesting to note that Annex 1 actually encourages the use of rapid microbiological methods within the Cleanroom Classification System, or CCS. This shows a clear regulatory push towards faster, more efficient microbiological testing in sterile manufacturing environments. Overall, these frameworks and requirements are designed to support the integration of rapid systems, helping us streamline quality control without compromising compliance or safety. Let's take a closer look at the comparative analysis. Here, we're really focusing on how different options or approaches measure up against each other. The key points to remember are the criteria we use for comparison, such as efficiency, cost, and scalability, and how each option performs in these areas. It's important to highlight that understanding these differences helps us make more informed decisions aligned with our overall goals. Connecting this back to the broader theme, this analysis emphasizes the importance of thorough evaluation in developing effective strategies. By comparing these elements side by side, we gain clarity on what works best and where improvements are needed. This step is crucial in advancing our project towards success. Let's take a closer look at the validation requirements, starting with the basics that apply to all methods. First, we need to ensure method suitability testing is in place. This step confirms that the method is appropriate for the specific application we're testing. Next, growth promotion testing is essential. It verifies that our media can support microbial growth, which is critical for accurate detection. And of course, media sterility testing ensures that our media are free from contaminants that could skew results. Now, when we talk about rapid methods, there are additional validation parameters to consider. Accuracy and precision are fundamental. They tell us whether the method provides reliable and consistent results. The limit of detection, or LOD, is also crucial, as it defines the smallest amount of microorganism our method can reliably detect. Furthermore, we need to perform equivalence studies, especially when comparing rapid methods to traditional methods, and this is where USP 71 comes into play. These studies help us demonstrate that the new rapid method is equivalent in performance to the established standard, ensuring confidence in its results. All these validation steps are interconnected and vital for ensuring our microbial testing methods are robust, reliable, and compliant with regulatory standards, ultimately supporting the safety and quality of our products. So, in conclusion, membrane filtration remains the gold standard and the preferred method for sterility testing because of its reliability and widespread acceptance. However, direct inoculation is still a conditional alternative, especially in situations where membrane filtration might not be feasible or optimal. Looking ahead, rapid systems are emerging as the future of sterility testing, offering faster results and improved efficiency. In terms of regulatory trends, there's a clear shift toward more rapid, automated, and risk-based approaches, which align with the industry's need for quicker decision-making and enhanced safety. The key insight here is that a hybrid strategy, combining traditional compendial methods with rapid techniques, is currently the best practice. This approach maximizes reliability while also leveraging the benefits of speed and automation, ultimately supporting better quality control in our industry. So, embracing this hybrid model helps us stay aligned with regulatory expectations and prepares us for the future of sterility testing. For further support, please don't hesitate to reach out to us. We're here to help you with any questions or additional information you might need. Just contact us through the provided channels, and we'll be happy to assist. Thank you so much for your time and attention today. We truly appreciate your engagement and look forward to supporting you in your journey.